Hello, this is Alex.
Hello, this is Joe.
Before we get into the process of mitosis, we need to know the cell parts. Here we have the cell membrane in green, and what's the white?
The white part is going to represent the nuclear envelope.
That's right. And here is the nucleus, and inside the nuclear envelope we have 4 sets of chromosomes, 2 in purple, 2 in red.
And there are the centrioles, and the yellow represents the microtubules.
They kind of look like whiskers. So we know that this cell is in prophase since we can see the chromosomes. Here are the purple chromosomes, thanks to Joe, and there are 2 of the same kind. And why is that, Joe?
Because they are called sister chromatids.
And why are they called sisters?
Because they are exactly alike.
During the duplication process of interphase, which the cell went, there are 2 chromosomes that will be preparing to split later on. Now, tell me more about these chromosomes. What do they have?
They have centrioles, which I am pointing at.
Centromeres, you mean?
Centromeres, which I am pointing at in the video, and they are located in the center of the chromosome.
Now, are they all located in the center?
Yes, I don't think so.
So in late prophase, why are you removing the nucleus?
Because the nuclear envelope is going to start to break up.
Now, I see that you're moving the centrioles and the microtubules to the side. How come?
Because new microtubules become assembled, and they need to move on opposite sides of the cell.
And then I'm guessing that the spindle fibers are... No, those aren't spindle fibers. Those are still microtubules that are being added to them. Am I right?
Yes, you're correct. So like I said before, Alex, in this phase right here, I will begin to cut up the nuclear envelope, and representing it, I will cut it with scissors every couple of inches.
So during metaphase...
You mean prometaphase?
How come?
Because the nuclear envelope is going to break up completely into numerous tiny flattened vesicles.
That's right. This would allow the chromosomes to be interacted by the microtubules that would be forming from the spindle. So what Joe is showing us here is the microtubules from both poles harness each chromosome and start pulling on it.
That's right. This will allow the microtubules to attach to the sister chromatids, which will be preparing for the next step, which is metaphase. During metaphase, the spindle fibers, represented by the sky blue color, will attach to one of the sister chromatids of the chromosomes.
So basically the job of the spindle fiber is to reel in the duplicated chromosomes to the midway point. We call this metaphase because as it goes into the poles of the completed spindle, it means midway between. This alignment is crucial for the next stage of mitosis. Joe, would you please explain why there's only 3 spindle fibers at one centriole, however there are 4 at the other?
Well, this is because there was a malfunction in the microtubules.
Now, is this normal?
No, this is not normal at all. Once all chromosomes are aligned in the middle, This starts anaphase. Each spindle fiber will reel in the sister chromatid that they were previously attached to at the centromere, or at the center of the centriole.
Once each chromatid is separated from its sister, this starts telophase. As the chromatid is separated from its sister, it has a new name, a chromosome. During telophase, it creates a cleavage furrow, which is sort of like an indent in between the cell. The baby blue string represents the contractile ring, which separates the two new cells that would be forming later on.
The chromosomes are no longer harnessed to the microtubules. They return to thread-like form. Vesicles derived from the old nuclear envelope fuse and form patches of membrane around the chromosomes. Patch joins with patch, and soon a new nuclear envelope separates each cluster of chromosomes from the cytoplasm.
Telophase is still not complete because cytokinesis, the process in which the cell actually divides into two, must occur.
The contrail ring will eventually pinch the cell until it separates it into two independent daughter cells.
Telophase, however, is still not over yet. We need two nuclei to form in the daughter cells. Finally, the end product of mitosis is two daughter cells. Each is diploid, which means its nucleus has two or each type of chromosome, just like the parent cell.
So we've taken you through the steps of mitosis, starting with prophase, the transition to metaphase, anaphase, and last but not least, telophase.
That's right.